Good evening and welcome to Monobiology. And this evening's lesson is on enzymes. And we've already talked about these in class. Enzymes are life's efficiency molecules. That's because they are involved in every single chemical reaction that happens inside of our bodies. But more importantly, they speed up the chemical reactions. They are, in fact, biological catalysts. And catalysts, recall, speed up chemical reactions without actually getting used up. More importantly, they lower the energy of activation for a particular reaction. Just a couple of things to refresh our memory on. Remember that enzymes are proteins, and they are three-dimensional proteins, which indicates that they are in the tertiary structure of proteins. And because they're tertiary structure, they have a specific shape. An important part to remember about their specific shape is that they form an active site. And that active site can fit a very particular reactant known as a substrate, which fits very nicely in here. When the enzyme attaches to the substrate, it causes certain things to happen that lower the energy of activation and allow the substrate to form new products, as you can see here. Okay, so that's just a refresher. What we're going to actually talk about today is how the cell or the organism regulates these enzymes. Because you can probably imagine if these enzymes were allowed to go and do whatever they wanted, they would just keep going and going and going. Because remember, they speed up chemical reactions, but they don't get used up. So there has to be certain ways to tell them to stop, and in some cases, maybe even to tell them to start. So that's what we're going to look at today. As I said, regulation is important for two reasons. First, it allows the cells or the organism to conserve energy. Second, it ensures that only the appropriate amount of products are produced. This is very significant to cells, especially individual cells who have limited resources, but it's also significant to multicelled, larger organisms who still, even though they're very large and have access to large amounts of resources, still have to have a conservatory mind. They have to conserve their resources. Now, these regulation of enzymes occur in two different places: during the reaction and before the reaction. And when I say the reaction, I mean the enzyme-substrate reaction. So let's have a look at the picture located here on this page to talk about the ones during the reaction. Okay? So what we have right here in this picture is the normal enzyme substrate reaction. Notice the substrate is going to bind to the active site on the enzyme. Simple. But what happens if the cell doesn't want that reaction to happen anymore? Well, there's a couple of things that it can do. Number one, it can introduce a process called competitive inhibition. And competitive inhibition essentially causes another substance that's not the substrate to bind to the active site. And once it binds to the active site, it outcompetes the substrate for that spot. If the substrate can't get to the active site, then no reaction will occur. The second type that occurs during the reaction is non-competitive inhibition. In this type of inhibition, a non-competitive inhibitor binds to a site other than the active site, and notice what happens right here to the enzyme. The enzyme's shape changes after the non-competitive inhibitor binds to it. Once this change occurs, the substrate can no longer bind to that enzyme. Again, cells have to have control of these enzymes, otherwise these enzymes are going to kind of just do whatever they want, and they will continue to use up resources, and they will continue to use up energy because they themselves are not used up in these chemical reactions. Let's talk about what happens in terms of regulation before the reactions. Regulation of enzyme activity often occurs before any reaction would even happen. In other words, the cell wants to regulate these enzymes. It wants to have them available, already put together, but not active. It only wants them active when it needs them to be active. Or, in some cases, it wants them active until it doesn't want them active anymore. And we're going to talk about those two situations. Generally speaking, the control of enzymes that occurs before the reaction is due to what we call allosteric activators or allosteric inhibitors. Now, what allosteric means is that it's a chemical that binds to a site other than the active site. Okay, so allosteric is a site other than the active site, and these regulatory mechanisms, the activators and the inhibitors that occur before the reaction, are generally considered to be under allosteric regulation because they bind to something other than the active site. Okay, so take a look at the first set here. Look at this one first. This is the allosteric activator. So here we have an enzyme, and the enzyme is going to be introduced. The cell is going to say, you know what, enzyme, I want you to do something, and therefore I am going to activate you. That activator is going to bind to the enzyme. What it does is it stabilizes the enzyme's shape into its active form. It changes it slightly, makes it ready to do its work, and now the enzyme is good to go. Whatever this enzyme is doing, it's ready to go. But before that, this enzyme is just floating around in the cell, just waiting until the cell calls on it. The other type of allosteric control that a cell would regulate in terms of enzyme activity would be the inhibitor. Okay, so in the first case with the activator, the enzyme was inactive to begin with, and we made it active by giving it the activator. In the second case, the enzyme itself is inactive to start with, but it is put into a stabilized inactive form via the use of an inhibitor. And you can see here the inhibitor is binding. Okay, so essentially an enzyme can oscillate or move back and forth between an active state and an inactive state, which is located down here. This inactive state down here is unstable. In other words, it could very quickly turn back into the active state. So what this inhibitor does is it binds to the inactive state and makes it stable. Now that it's stable, it can't go back into its active form. Both of these things, the activators and the inhibitors, would be ways that the cell would be able to regulate the activity of the enzyme so that it would only be working when the cell wants it to be working. And that, in a nutshell, is how these particular things work. Please bring any questions you might have to class on Monday, and we will certainly have a discussion about it. Good evening.